In this presentation, we'll examine primary structure of nucleic acids. Nucleic acids are polynucleotides. They are polymers composed of nucleotides. A nucleic acid is composed of a sugar phosphate backbone, and nucleic acid bases are attached to the backbone as side chains. Each nucleic acid has a unique sequence of bases, while sugar phosphate backbone is common to all of them. In RNA, bases are adenine, guanine, cytosine, and uracil, and of course they are attached to sugar phosphate backbone or ribose phosphate backbone. In DNA, bases are adenine, guanine, cytosine, and thymine, and sugar phosphate backbone is deoxyribose phosphate backbone. Here is schematic representation of sugar phosphate backbone and nucleic acid bases attached as side chains to it. Beta-N glycosidic bonds attach nucleic acid bases to sugar phosphate backbone. Here is an actual formula of DNA molecule. We know that this is DNA molecule because sugar is 2-deoxyribose. And we can see sugar phosphate backbone. We can see that it contains 3', 5' links as phosphodiester bridges. And of course, nucleic acid bases are side chains attached to it. You can see that bond shown in purple is beta-N glycosidic bond. Here are again segments of RNA molecule on the left. So you can see RNA molecule has ribose as sugar component and segment of DNA molecule on the right, 2-deoxyribose as component. And of course, RNA has uracil, while DNA contains thymine. We can represent DNA or RNA molecule with structural formulas as shown on the previous slides. But this is almost never done. The reason for it is that DNA and RNA molecules are very large polymers. They contain thousands or tens of thousands of nucleotide residues. And it would be very difficult to try to represent them with structural formulas, and also information content would be very low because there are only 4 different nucleic acid bases, and sugar phosphate backbone is common to all of them. So we resort to shorthand representation. The most common shorthand representation utilizes single capital letter to indicate each nucleotide residue. And one can insert lowercase letter p to indicate phosphate bridge. So that's shown here in this first row. A P preceding the sequence, as in this case, indicates that phosphate group is at 5' end, because obviously chain is listed from left to right, from 5' to 3' end. If letter P, lowercase letter p, terminates the sequence, that indicates that phosphate group is at 3' end. Since sugar phosphate backbone is common to all DNA and RNA molecules, we usually omit lowercase p as phosphate bridge and simply understand that it's there. We understand that sugar phosphate bridge is there, and we simply omit lowercase p. We only may want to use that one to indicate that phosphate is in position 5', as shown in the 4th row from the top, or 3' position, as shown in the last 5th row. Finally, we can specify that molecule is DNA molecule by inserting D for deoxy prior to the sequence. But that's usually not necessary, because from the composition one can determine that molecule is DNA, because DNA molecule contains thymine, so obviously indicated as T, while RNA molecule contains uracil, indicated as U. So this completes our brief overview of primary structure of nucleic acids, and next we are going to look into secondary structure.